Hello, welcome back to Biology with Risa. This is part 3 of my 5-part lecture series on the male and female reproductive system. In this video, we will look at gametogenesis, or in other words, the production of sperm and eggs. Gametogenesis is the process of forming gametes. Another word for gametes are sex cells. And this primarily uses a type of cell division called meiosis. There are 2 types of gametogenesis. Spermatogenesis is the process of producing sperm, or in other words, spermatozoa. Spermatogenesis happens within the seminiferous tubules of the testes. Oogenesis is the process of producing eggs, and another technical term for eggs are secondary oocytes, and this happens within the ovaries. In order to understand the steps involved in gametogenesis, you have to have a basic understanding of the 2 types of cell division that occur in living organisms. Cell division is important in making new cells. One type of cell division is called mitosis. This produces new cells that are genetically identical to the original cell. So let's look inside of our bodies for an example. All the cells inside your body except your sperm or eggs have 46 chromosomes or strands of DNA. So if one of these cells undergoes mitosis, it's going to replicate its DNA and then split the DNA into 2 separate cells, each cell containing 46 chromosomes, each cell identical to the original cell. When might this need to happen in our bodies? Well, this happens during growth. So you start out life as a single cell. So this is actually showing a zygote. This is the result of fertilization of a sperm and egg. You can actually see the nucleus of the sperm and nucleus of the egg fusing here. So this cell would have 46 chromosomes in it, and that's how you begin your life. This one cell will divide into 2 cells. Those 2 cells divide into 4 cells. And then those 4 cells divide into 8 cells, and they just keep multiplying until it forms you, and eventually you're made up of 37 trillion cells that are genetically identical to each other. This mitosis is also important in the repairing of tissues. So if tissue becomes damaged, then in order to repair those cells must undergo mitosis to replicate themselves to create genetic copies. Furthermore, you're constantly replacing cells in your body. For example, you're constantly replacing the cells lining your stomach. You're constantly replacing your skin cells. In order to replace those cells, new cells are made through mitosis. So when does meiosis occur? Meiosis only occurs, so the only purpose of meiosis is to produce sex cells, or in other words, gametes, the sperm and eggs. What meiosis does is 2 very important things. So this type of cell division, first off, halves the number of chromosomes. So this makes sure that you only give half of your genetic information to your offspring. In addition, it creates genetic variation. So it mixes up the DNA so that the chromosomes are not exactly identical to what's inside your body. And so this creates some genetic variation that can be passed on to your offspring as well. So again, meiosis halves the number of chromosomes and it creates genetic variation. Therefore, since meiosis halves the number of chromosomes, it makes diploid cells, or cells with 2 sets of chromosomes, haploid. And so they only have 1 set of chromosomes. So let me show you what this means again. So every cell in your body except for your gametes are diploid. They actually have 2 sets of chromosomes, 46 total, or in other words, 23 pairs. So you got 23 chromosomes from one parent and 23 chromosomes from the other parent, and that makes a diploid cell. So here you can see the 23 pairs of chromosomes that would be present in a normal diploid cell. It has 2 sets of chromosomes, 1 set from each parent. However, meiosis takes a diploid cell and it halves the number of chromosomes. So now those cells are considered haploid and they only have 23 total chromosomes. So 1 of the chromosomes from each pair is removed. So now there's only 23 total there. So meiosis always goes from diploid to haploid. Mitosis, which we talked about in the last slide, always goes from diploid to diploid in humans because you're creating a clone of the original cell. So again, meiosis is how sperm and eggs are made. So within the testes, in males, they have these diploid cells called germ cells. Germ cells is just a general term for cells that will eventually become gametes. So these germ cells have 46 chromosomes. They'll undergo meiosis to create haploid sperm, and those haploid sperm will each have 23 chromosomes. In females, you have diploid germ cells in the ovaries. These have 46 chromosomes. They're going to undergo meiosis to create a haploid cell with half the number of chromosomes, so 23 chromosomes. And those haploid cells are called eggs or oocytes. So let's look here to further differentiate between mitosis versus meiosis. Now mitosis, again, if we start with 46 chromosomes, then the two resulting cells will be identical and each have 46 chromosomes. Notice mitosis only undergoes one round of division. You start off with one cell and then it splits to create two cells. Meiosis is a little bit different. It requires two rounds of division. So you start off with the cell with 46 chromosomes, it's diploid, and it splits into two cells. And the function of meiosis 1, or the purpose of meiosis 1, is to reduce the chromosome number in half, also to allow for some shuffling of the genes which occurs. So some of the DNA is swapped between the chromosomes. And so what the result of meiosis 1 is two haploid cells. However, these two haploid cells have copies of the DNA inside of them, so they actually have to be further divided again. And we're not going to get into the details of this in this lecture. However, just know that meiosis 2 has to occur, and these two cells here will further divide into, each will divide into two cells. So the result is 4 genetically different cells. Each one is haploid with 23 chromosomes. So again, meiosis starts with one cell. Meiosis 1 results in two cells, and then meiosis 2 results in those two cells dividing. So we end up with 4 daughter cells. Each one is haploid. Each one is genetically different from each other. Okay, so those are the big differences between mitosis and meiosis. So when does mitosis and meiosis occur again in the human life cycle? Well, again, you start off life as a diploid zygote with 46 chromosomes. And a lot of times we reference diploid by using this abbreviation 2N because it means two sets. Okay, so you have two sets of DNA totaling 46 chromosomes. This diploid zygote undergoes mitosis, and that creates you. You are a multicellular organism with 37 trillion cells or more or less. And each one of those cells has 46 chromosomes in it. The only time meiosis occurs is within the ovaries or the testes to make haploid eggs or haploid sperm, each containing 23 chromosomes. The haploid sperm and the haploid egg will fuse together through a process called fertilization. So fertilization is when two haploid sex cells, each with 23 chromosomes, fuse to form a single diploid cell. This diploid cell is called the zygote, and it contains 46 chromosomes. From that one zygote, you create an entire new human. So now let's look at the specifics of spermatogenesis and a process called spermiogenesis, which we'll talk about in a little bit. So again, spermatogenesis is the process that produces sperm. It occurs in the seminiferous tubules of the testes, and it begins at puberty and is continuous through a man's life. So here is a small section of a seminiferous tubule. This is the periphery of the seminiferous tubule, and this is the lumen of the seminiferous tubule, or the space in the inside of the tube. Notice on the periphery, we have a germ cell called a spermatogonia, and it's going to undergo cell division to create what's called a primary spermatocyte. This divides to create a secondary spermatocyte, and then the secondary spermatocytes produce spermatids. And then the spermatids will grow a tail, and they're called spermatozoan or sperm at that point. Now, there are a couple hormones that help to regulate sperm production, and you want to know the role of FSH and LH in sperm production. Now, remember, the hypothalamus secretes a hormone called gonadotropin-releasing hormone. This stimulates the anterior pituitary to release FSH and LH. FSH and LH have slightly different roles in sperm production. LH, also known as luteinizing hormone, stimulates the interstitial or Leydig cells within the testes to make testosterone. Remember, the interstitial or Leydig cells are cells that are between the seminiferous tubules, so they are outside of the tubules itself. LH in males is sometimes also called interstitial cell stimulating hormone, but I will just refer to it as luteinizing hormone because it's the same hormone that's produced in females. The other hormone that the anterior pituitary releases is called follicle stimulating hormone, or FSH. And this actually stimulates the sustentacular cells, also known as the Sertoli cells, to release a protein called androgen binding protein. And this protein binds to testosterone to make testosterone more effective. So androgen binding protein in conjunction with testosterone are both needed to stimulate spermatogenesis. So let's begin to look at the actual process of spermatogenesis. Spermatogenesis begins with those germ cells called spermatogonia, and these are diploid cells, again, in the periphery of the seminiferous tubules. And these spermatogonia, or germ cells, can actually divide or clone themselves through mitosis, and this provides a lifetime supply of spermatogonia in males and is why they can produce sperm their entire life. So here you can see a spermatogonia. It underwent mitosis to create more spermatogonia. Some of these spermatogonia will actually develop into sperm. And the developing sperm, as they go through the several stages of development, they will move towards the lumen of the seminiferous tubules. And you can see that the spermatogonia produce primary spermatocytes. These primary spermatocytes are going to undergo meiosis 1, first round of meiosis, and produce secondary spermatocytes. They will undergo meiosis 2 to produce spermatids. Spermatids are cells that are very much like sperm, but they don't have a tail yet. So they have to grow their tail, and when they grow their tail, it's called spermiogenesis. And then we have a spermatozoa, and that can then make its way to the epididymis to further mature. So now let's summarize the steps involved in spermatogenesis. I recommend studying this slide over and over again, and even trying to just draw out this flow chart by yourself. Okay, so we start off with the spermatogonia. These are the germ cells, and remember, they're diploid. They have 46 chromosomes. Some of the spermatogonia will undergo mitosis just to create more spermatogonia, so this is what creates the lifetime supply of sperm in males. And some of the spermatogonia will undergo mitosis to create what's called a primary spermatocyte. The primary spermatocyte also has 46 chromosomes, and that makes sense because mitosis just clones the original cell. Now, the primary spermatocytes are destined now to become sperm. They're going to undergo meiosis 1, so each primary spermatocyte will divide into secondary spermatocytes. And as a result of meiosis 1, we go from being diploid to haploid already. Those 2 secondary spermatocytes are going to each undergo meiosis 2, a second round of division, to create a total of 4 spermatids. Spermatids are kind of very similar to sperm, except that they do not have their tails yet. So the process in which they grow their tails and undergo some other morphological changes is called spermiogenesis. Spermiogenesis of the spermatids results in 4 spermatozoa, also known as sperm. Each one is haploid and each one now has a tail and can make its way to the epididymis, where it will further mature for a couple of days. Now, it is really important not to confuse the term spermatogenesis with the word spermiogenesis. So let me define both of these words to make sure that you know the difference. Spermatogenesis is the entire process that produces sperm. So it begins with the spermatogonium and ends with the spermatozoa. It includes mitosis and meiosis and spermiogenesis, all the processes necessary for that process to happen. Spermiogenesis is just the last stage of spermatogenesis. So it's where the spermatids become spermatozoa. That is spermiogenesis. The spermatozoa, again, once they form, they're going to leave the seminiferous tubules and they're going to go to the epididymis where they will mature. It actually takes 64 days for a spermatogonium to develop and become a functional mature sperm. So let's look a little bit more of this at this process of spermiogenesis. So here we have a spermatid, and you can see that it doesn't really have a tail. It has a lot of cytoplasm in it. One thing that's going to happen is that it's going to start to grow a flagellum or tail here. It's going to lose all its excess cytoplasm. And eventually it's just going to create a mature sperm with a head, a mid-piece here, and a long tail. Despite having a tail after spermiogenesis, however, I want to remind you that the sperm are not capable of swimming until they mature in the epididymis. And even though they are capable of swimming after maturing in the epididymis, they will not actually swim at all in the male reproductive tract. Sperm do not swim until they enter the female reproductive tract. Let's look a little bit at the anatomy of a sperm cell or spermatozoan. So they consist of 3 parts. We have the head here, the mid-piece here, and then the tail at the end. The head consists simply of, mostly it contains just the nucleus, so the genetic material. And then there's a little cap, a little vesicle or structure that forms here called the acrosome. The acrosome is extremely, extremely important because it contains digestive enzymes, and these digestive enzymes will be released when the sperm contacts the egg, and it will digest the external covering of the female gamete so that the sperm can actually penetrate the egg. The mid-piece of the sperm simply contains a whole bunch of mitochondria. And if you remember, mitochondria make ATP, so this is supplying the energy that the sperm needs to swim once it's inside the female's reproductive tract. And then, of course, we have the tail, which provides motility. Now let's look at oogenesis. I recommend going back and reviewing spermatogenesis and remembering the steps before you look at oogenesis because they're going to be very similar in the steps that are involved. So oogenesis is the production of an egg, also known as an ovum or oocyte, within the ovaries. It also begins with a diploid germ cell containing 46 chromosomes. However, this cell is called an oogonia instead of a spermatogonia. Oogonia form during fetal development and become primary oocytes before birth. The primary oocytes, however, are arrested in meiosis 1 until puberty. So a lot of people have the misconception that we're born with all the eggs that we're ever going to have. Well, these aren't really truly eggs yet because they haven't completely gone through all the steps of oogenesis when you're born. However, you are born with cells that have already began the process of oogenesis. So let's look at the role of FSH and LH in females and in the process of oogenesis. So this differs a little bit than in males because FSH and LH levels in females undergo cyclic changes in hormone secretion that occur monthly, which coincide with our monthly menstrual cycles. FSH is going to stimulate the follicles to develop within the ovaries. And remember that each follicle contains a developing oocyte. So FSH triggers these follicles to develop and mature. and they allow oogenesis to kind of continue. During your monthly cycle, there will be a period halfway through the cycle, so on day 14, where there will be a large spike in the luteinizing hormone. And when this large spike happens, that triggers ovulation, or the release of the oocyte from the ovaries into the uterus. LH will also, after this spike happens, LH will also simultaneously trigger the follicle, the remaining follicle cells within the ovaries, to develop into what's called a corpus luteum. And this secretes a whole bunch of progesterone after ovulation. This progesterone is going to be really important in kind of thickening the uterine lining and creating an environment capable of sustaining or feeding the implanted embryo. However, I will review the role of FSH and LH in the female reproductive cycle again in the next lecture video. So if this seems a little overwhelming right now, remembering what these hormones do, we will go over it again in the future. Okay, so let's look at the steps involved in oogenesis and see if we can pick out some similarities between oogenesis and spermatogenesis, and also see if we can pick out some differences. So remember, spermatogenesis began at puberty. And this is a little bit different than in oogenesis because everything that's happening above this line in this diagram is happening before the female is even born. So before the female is even born, she has a whole bunch of oogonia. They undergo mitosis. Some of them form more oogonia, and some of them, the oogonia, become primary oocytes. Okay, so that was similar to as in males. The primary oocytes will then undergo meiosis 1. However, they'll start to go through cell division and then they'll stop. They'll pause at the beginning of meiosis 1 in a stage called prophase 1. So before birth, you're born, if you're female, with a whole bunch of primary oocytes inside of your ovaries, and they're arrested until you go through puberty. Once you go through puberty, some of these primary oocytes every single month will start to complete meiosis 1, and they will become a secondary oocyte. However, notice that when this primary oocyte divides, it doesn't create two secondary oocytes that are equal in size. Instead, one of the cells pretty much steals all the cytoplasm, and then the other cell, which we call a polar body, is really tiny and just contains genetic information, and it doesn't have a lot of cytoplasm. It will eventually die. So the polar bodies will die. Their only function is really to help take away some of the genetic information. That secondary oocyte will then be ovulated. And so this is what's actually ovulated from the ovaries. And they still haven't undergone meiosis 2 yet. And the secondary oocyte will actually never undergo meiosis 2 unless a sperm begins to penetrate its outer membrane. As soon as a sperm starts to penetrate the outer membrane, then that will trigger that secondary oocyte to undergo meiosis 2, and it will then become a mature ovum. And that mature ovum can then be fertilized completely by the sperm to create that zygote we talked about before. So again, let me review this again. We have the oogonium. It undergoes mitosis to create primary oocytes. These primary oocytes are arrested in development until puberty. Then during puberty, every month, some of these will become secondary oocytes. The secondary oocytes are what are ovulated from the ovaries. And if fertilization doesn't happen, the secondary oocyte will just die. The only time the secondary oocyte will complete meiosis 2 is if fertilization happens from a sperm. And then it will complete that last round of division. Okay, so some important observations to make here. The process is similar to spermatogenesis, but the timing of the events differ. Also, another really important point here is that only one gamete survives during oogenesis, whereas in males, there were 4 gametes that resulted from spermatogenesis. In females, only 1 mature egg is made. During oogenesis, it's essential for the oocyte that's going to become the ovum to have most of the cytoplasm, because it's going to have to supply nutrients to the developing zygote during the period between fertilization and implantation in the uterus, because it takes a couple of days before the actual fertilized egg will implant into the uterine lining. Also in females, remember that meiosis 2 never finishes unless the sperm penetrates the eggs. So in other words, a mature ovum only forms if fertilization occurs. So this is a diagram that I've made to actually help to visually see the steps involved in oogenesis. Again, I recommend drawing this out yourself multiple times to remember the major steps involved here. So we start with oogonia. This is before birth. Oogonia are diploid. They have 46 chromosomes. They undergo mitosis to create the primary oocytes, which also have 46 chromosomes. It's the primary oocytes that undergo meiosis 1 to create the secondary oocyte, which has only 23 chromosomes, because meiosis will reduce the chromosome number by a half. Only when this cell splits, it creates 1 secondary oocyte, and the other cell is called a polar body that will eventually just die. This secondary oocyte will undergo meiosis 2, but only if it's penetrated by a sperm cell. If the sperm cell penetrates it, it will become a mature ovum, and then it will also form a polar body that dies. And then that mature ovum, now that it's fertilized, will create a zygote with 46 chromosomes, because the 23 chromosomes from the ovum and the 23 chromosomes from the sperm will combine together to create a new diploid cell. So within the ovaries, there are actually 2 processes that are happening simultaneously. So as oogenesis is happening, the cells that are surrounding the developing oocyte are also maturing. And if you recall, the ovaries contain many follicles, and these are spheres of cells that contain a developing oocyte. As the oocyte develops, these follicles begin to mature and grow, and this process is called folliculogenesis. It takes about 290 days to complete the entire maturation process, which means that you constantly have follicles maturing inside of your ovaries. So let's look at the order that these follicles develop. So it begins with what is called primordial follicles, and these actually are found on the periphery of the ovaries, and you can see them as these little dots out here. You were born with many of these primordial follicles, and each one contains a primary oocyte, because remember, you're born with the primary oocytes. These primordial follicles can stay in a resting state for years, and each month a few of them will start to mature. So maybe up to like 30 will mature each month. The primordial follicles will mature, and they will become what are called primary follicles. Primary follicles have a larger oocyte inside, and the follicle cells surrounding it, or follicular cells surrounding it, form a single layer around the oocyte. The primary follicles then become secondary follicles. These have even a larger oocyte, and the follicle cells now start to form layers on the outside there. You may also start to see a fluid chamber or space beginning to form within the follicle called an antrum. The secondary follicle then becomes a tertiary follicle, and you can see the antrum starts to get bigger and fill with more fluid. Also, the layers of the follicle cells start to become more and more. Each month, only one of these tertiary follicles will mature and become a mature follicle. So again, only one tertiary follicle from each month's cohort will mature and become what we call a mature or Graafian follicle. And this mature follicle will contain a secondary oocyte, and that secondary oocyte will be released during ovulation. The remaining maturing follicles or tertiary follicles that were maturing at the same time will actually just degenerate and die. So to put things kind of into perspective, by birth, females are born with about 2 million primordial follicles. So you can see these all on the outside of the ovaries here. Each one of these, remember, contains a primary oocyte. However, these primordial follicles are constantly dying. And so by puberty, most of the primordial follicles have degenerated. So by the time you reach puberty, there's only about 200,000 to 400,000 of these primordial follicles that remain. After puberty, the number continues to decline until a female reaches menopause. And at that point, there's pretty much no follicles left within her ovaries. So now let's look a little bit at what happens after ovulation. So remember again, we start with the primordial follicles. They become primary follicles. Then they mature into secondary follicles and then into tertiary follicles. Only one of these tertiary follicles each month will become a mature follicle or Graafian follicle. That mature follicle will release a secondary oocyte during ovulation. However, notice that some of the follicular cells stay behind whenever it releases a secondary oocyte. Those remaining follicular cells will form a hormone-secreting structure called the corpus luteum. The corpus luteum is a remnant of the mature Graafian follicle after ovulation. And it has a very, 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 very important role in female reproduction because it begins to secrete high amounts of progesterone and also small amounts of estrogen. If fertilization occurs, the progesterone levels will continue to rise. This will stimulate buildup of the uterine wall. It will increase vascularization and increase secretions of the uterine wall, and that prepares the uterus for implantation. However, if fertilization doesn't occur, which is more typical every month, if there's no fertilization that occurs, then the secondary oocyte dies and the corpus luteum breaks down to form a little scar in the ovaries called the corpus albicans. Because the corpus luteum has degenerated, progesterone levels fall. And it's this fall in progesterone levels that triggers a female's body to undergo menstruation. And instead of the uterine lining continuing to build, it actually sheds the uterine lining. So the reason that a woman menstruates is because of a drop in the progesterone levels as the corpus luteum degenerates. So let's look at the steps again of folliculogenesis now. So this is again another diagram that's really good to just draw out over and over again to study the steps that are involved. We start with primordial follicles. What do they become? They become primary follicles. They mature into secondary follicles. Secondary follicles mature into tertiary follicles. Tertiary follicles mature into what we call mature follicles or a Graafian follicle. And remember, only one tertiary follicle will mature completely. Any other tertiary follicles that are around will degenerate. Then what happens is the mature follicle will release a secondary oocyte through the process of ovulation. And that will then form in the ovaries a corpus luteum. If no pregnancy occurs, or in other words, no fertilization occurs, what happens to the corpus luteum? It degenerates and it forms what's called the corpus albicans. All right, so now that we've looked at both spermatogenesis and oogenesis, let's compare them side by side. Both of these begin with germ cells that are diploid. In spermatogenesis, it's called the spermatogonia. In oogenesis, it's called the oogonia. These cells undergo mitosis to create either the primary spermatocyte or the primary oocyte. The primaries undergo meiosis 1 to form the secondary spermatocytes or the secondary oocytes. Now the difference here though is in females, we only get one secondary oocyte and the other cell is called a polar body, which degenerates and dies. The secondaries undergo meiosis 2. In males, this forms 4 spermatids. Each spermatid then undergoes spermiogenesis to grow a tail. And this results in 4 functional sperm cells. In females, That secondary oocyte will only undergo meiosis II if a sperm begins to penetrate its cell membrane. Then it will become a mature ovum, which can then be fertilized, and that creates a zygote. So a major difference here that we see is oogenesis only makes one mature egg through its process, and it also creates those polar bodies, which are non-functional cells that die. Meiosis I is what creates haploid cells. So the germ cells and the primary oocyte and spermatocyte are diploid. Everything after is haploid, except for, of course, the zygote. This table further compares spermatogenesis and oogenesis. So where does spermatogenesis occur? It occurs in the seminiferous tubules in the testes. Oogenesis occurs in the ovaries. Now let's look at the number of gametes produced during a lifetime. In males, there's lifelong production, and the numbers can reach as much as 400 billion sperm per lifetime. Whereas in females can only produce a fixed amount of eggs, and that's about 400 that mature and are ovulated in their entire lifetime. So women tend to go through about 400 menstrual cycles their entire life. How about the number of gametes produced per germ cell? For spermatogenesis, how many gametes can they make per spermatogonia? That would be 4. And oogenesis, how many gametes can they make per oogonia? That would just be 1. When does the process of spermatogenesis begin in males? That would be puberty. What about in females? When does oogenesis begin? Well, that would be during fetal development, so before birth. But remember, it's arrested in meiosis I until puberty. What about the timing of gamete release? For spermatogenesis, it's any time. In oogenesis, however, it's only monthly, so it has to follow the menstrual cycle. Does it ever end? Spermatogenesis doesn't end, however, fertility may be reduced as males get older. Oogenesis, on the other hand, does stop eventually. Fertility stops when women reach menopause, and this occurs again because there aren't any follicles left inside the ovaries. Are meiotic divisions continuous in spermatogenesis? Yes. In oogenesis, no. They pause several times. For example, they're arrested in meiosis I until puberty, so you're born with only those primary oocytes, and then after puberty, they start to become secondary oocytes, which are ovulated. And then the secondary oocytes never complete meiosis II unless fertilization occurs. All right, so here are some review questions. What is the difference between spermatogenesis and spermiogenesis? Remember, spermatogenesis is the production of sperm. It's the entire process going from spermatogonia all the way to spermatozoa. Whereas spermiogenesis is just referring to the process where we go from spermatids to spermatozoa. So when the spermatids kind of grow their tail and get rid of excess cytoplasm and so forth. Spermiogenesis is the last stage of spermatogenesis. What is the chromosome number of the following cells? So these cells are listed in order of spermatogenesis. How many chromosomes are in each one? You can summarize it simply as the first 2 here are diploid, have 46 chromosomes, because spermatogonia produce primary spermatocytes through mitosis. The rest will be haploid and only have 23 chromosomes. What is the chromosome number of the following cells here? Well, the beginning is the same as spermatogenesis. These cells are diploid. The oogonia produce primary oocytes through mitosis. So they are diploid with 46 chromosomes. The secondary oocyte, however, is haploid. It has 23 chromosomes because it is the result of meiosis. However, the zygote is diploid because the zygote is the result of what process? Fertilization, where the sperm and the egg fuse. All right, so that is the end of video 3. To learn more about reproduction, watch video 4, which looks at the hormones involved in the female reproductive cycle.